So for case 2, it talks about regulation of glucose uptake by the cell. The question asks, in normal tissue, how is glucose uptake regulated? And it gives me a list of different tissues here. We have the liver, skeletal muscle, brain, and adipose tissue. So in the liver, glucose is taken up via GLUT2 or GLUT2 receptors in the cell membrane. The GLUT2 receptor is insulin independent. So glucose is able to readily enter the cell, and inside of hepatocytes or liver cells, glucose can then be used for glycolysis, gluconeogenesis, or fatty acid synthesis. The next tissue lists skeletal muscle, and skeletal muscle takes glucose up via GLUT4 receptors in the cell membrane, but GLUT4 receptors are insulin dependent receptors. Therefore, we need insulin acting on the receptor to cause that autophosphorylation, to create stimulate the entire cascade, which inevitably moves that GLUT4 protein to the cell membrane to allow glucose to enter the cell. Once glucose is able to enter the cell of skeletal muscles, it can then undergo glycolysis or glycogen synthesis. The next tissue is brain tissue, and brain tissue takes up glucose via the GLUT1 and GLUT3 receptors, and these are insulin independent receptors. Therefore, our brain tissue is able to get, or our brain cells are able to get glucose readily without any delays. And if I'm not mistaken, I believe our brain cells can only last about like 3 minutes without glucose before cellular death. So it's really important, and it makes sense why it has 2 different receptors on the membrane. In the brain cell, the glucose is going to undergo glycolysis. And the last tissue listed here is adipose tissue. And adipose tissue receives glucose or uptakes glucose via GLUT or GLUT4 receptors, which are insulin dependent, just like skeletal muscle. Therefore, insulin is necessary to allow the glucose to enter the cell. Inside of the adipose tissue, glucose is able to undergo glycolysis, and in states of excess glucose, the adipose tissue can undergo fatty acid synthesis. The next question asks, how does glucose uptake in normal tissue compare to glucose uptake by tumor cells in the above image? In the above image, it shows how there's a high amount of glucose utilization or uptake versus the other tissues. And the second part of the question says, why would this be advantageous to an invasive tumor? So to answer the first part of the question, how does this uptake in tumor cells differ than normal cells? The uptake of glucose in tumor cells drastically exceeds the normal glucose uptake in normal cells. In normal cells, our glucose is put through glycolysis and ran through the TCA cycle and ran through the electron transport chain. For tumor cells, they are primarily in environments with hypoxic conditions. So the tumor cell is less inclined to utilize the TCA cycle or electron transport chain, but it still wants to create energy molecules for its cellular proliferation. So what the tumor cell undergoes is a large amount of glucose uptake and further catalyzation of pyruvate into lactate. So it utilizes lactic acid fermentation to get to replenish the NADs needed for glycolysis. Now, the reason why it does things that way were from what I mentioned before about the hypoxic conditions, but at the same time, if we remember, the pyruvate kinase M2 is working at a slower rate, so the tumor cell has much more glycolytic intermediates available, so it can utilize glucose at pretty much like it can put glucose through glycolysis at a higher frequency if you think about the intermediates available. And also, the second part of the question says, how would this be advantageous to an invasive tumor? So with the lactic acid fermentation occurring, this changes the tumor microenvironment in the surrounding tissues, causing more acidic pHs. And what acid can do to normal cells is it can decrease cellular function for normal cells. Cells in particular being cells of the immune system, like killer T cells and cells that are utilized, like white blood cells that can track down different tumors and end up programming cell death, things of that nature. The way I see the lactic acid fermentation altering the environment, I see that where I think of that as a smoke screen, pretty much, to kind of keep the tumor in an elusive environment where it can't really be found, because when cells enter that area that do that monitoring, their function becomes altered. So that is one of the ways that this lactic acid fermentation can help or be beneficial to an invading tumor. The last part of the question asks, what modifications may have occurred in cancer cells to alter glucose uptake as predicted above? So cancer cells undergo many modifications, but modifications that are most prevalent in relation to glucose uptake have to do with the oxygen state of the tumor itself. So with a tumor being primarily just a mass of cells, there's portions of the tumor that do not get a large volume of oxygen, if any. So the hypoxic tension is high, and when portions of a tumor experience hypoxic conditions, just like our normal cells, it can stimulate a gene transcription factor called hypoxia-inducible factor 1. And what hypoxia-inducible factor 1 does, which benefits the cancer cell, is it upregulates the gene transcription of enzymes which are utilized in glycolysis. So now this tumor cell is able to receive or produce more enzymes for glycolysis, more enzymes which are used in glycolysis, enzymes like hexokinase and pyruvate kinase or phosphofructokinase, things of that nature. Throughout the entire glycolysis cascade, it's now has more enzymes to move reactions forward and pretty much produce more cellular energy, more ATP. In addition to the hypoxia-inducible factor increasing the gene transcription of the enzymes for glycolytic activity, it also increases the production of the different receptors, GLUT1 and 3, the gene transcription factor increases the synthesis of GLUT1 and 3 receptors. So now the tumor cell is also able to have these receptors that readily let glucose enter the cell because they are insulin independent receptors. And that actually makes a lot of sense since we're looking at a brain. We talked about the brain, the receptors of brain tissue being GLUT1 and 3. So now this tumor is able to receive glucose without requiring any insulin. In addition to the receptors and the glycolytic enzyme synthesis, hypoxia-inducible factor elicits the activity of another factor which promotes angiogenesis to the tumor itself. So now the formation of blood vessels occurs around the tumor. So now the tumor has a direct source of glucose and nutrients to it. And this pretty much all promotes cellular proliferation, glycolysis, and it just really benefits the tumor cell drastically. So those are the modifications that can occur in a tumor cell that allow it to uptake glucose more readily. And also that concludes the case 1 and case 2 questions for our Unit 2 case connections video. So, all right, have a good one. Bye.